Hi everyone, this is lecture 32, Quantitative Traits. So far we've talked about Mendelian inheritance, for example, complete dominance, complete recessive, incomplete dominance, codominance, lethal alleles. But most traits, as you probably sort of realize, are more complex, and they don't have simple ratios like Mendel found. So we're going to start talking about some of the more complex kinds of ratios that you would see in real life. So this material comes from chapter 22, page 650 to 655. First quiz question here. When a trait has a continuous distribution, such as height, and most individuals in the population are near the average, the distribution of the trait is most likely a what kind of distribution? Number two, when several genes control a phenotype and each gene has an allele that contributes an equivalent small increment to that phenotype, we call that inheritance pattern what? In the early 1900s, when Mendel's work had been rediscovered, scientists were trying to figure out how to relate his principles to complex ratios. For example, the ones that would be seen in sort of the real world. For example, most traits we know do not produce simple ratios, and crosses of hybrids, as shown in this diagram here, produce what we call a normal distribution. For example, you have two populations, a dwarf population, a tall population. They're each true breeding. They generate a hybrid that's intermediate in height. But self-crossing the individuals in the F1 generation produces a range of heights, not a simple 3-to-1 ratio. This kind of variation is seen all over in nature, from shell patterns in snails, as shown here, where it's many genes that are controlling these patterns, to things like human health and human diseases, where diseases like cardiovascular disease or diabetes have many contributing alleles that can add to your risk or decrease your risk of developing that disease. Normal distributions look like this. They can have all the same average in this case. So there's an average, and the average has the most represented in that population. But there can be a range of variance, which is shown here. These S-squared numbers are the variance. And so different populations may have different variances and yet have the same average. With Mendelian traits, we've talked about discontinuous variation, for example, tall and dwarf pea plants, red eye flies, etc. But we're going to begin talking about continuous variation, the kind that fits to a normal distribution. For example, human height, human weight, again, risk for developing disease, things of that nature. There's also another kind of variation called meristic. Meristic traits are those that have a distribution, yet the phenotypes are defined by whole numbers. For example, the number of seeds in a pod, the number of eggs laid. You can't have a fraction of an egg laid by a chicken, for example. But you can be a fraction of a foot tall. As a model for understanding continuous variation, we're going to go back to incomplete dominance. This is a very simple model for understanding how this principle works on a larger scale. With incomplete dominance, as seen here with the snapdragons, you have parents who are true breeding, either red or white. They produce an intermediate in the phenotype in the F1 generation, which is pink. Crossing the two pinks together gives you a 1 to 2 to 1 ratio. Instead of showing this as a diagram like this, we can represent this as a graph. And if it was represented as a graph, we could graph it like this, where the F2 phenotypes are on the x-axis and the frequency of seeing those phenotypes is on the y-axis. The reds in the F2 generation are 1/4 of the total offspring, so hence 25%. And there's one of those. The pinks is 50% of the total offspring, whereas the whites, again, is 25% of the offspring. And so we can graph this from left to right, red to white, sort of a decrease in red color from left to right across the screen. And again, this is one gene pair where if you use the capital letter A to represent red and the lowercase a to represent white, you would generate red plants that are AA, big A, and white plants that are little a, and of course the pink plants would be big A, little a. Well, one of the scientists that was working in the early 1900s was this French scientist, Nielsen L. And he studied wheat, and very similar to the snapdragons, the wheat had sort of a reddish tinge to it. And there was the red varieties, and then they went down to white varieties. So you would have everything in between. And so when he did this kind of study, he would find a red strain, he'd cross it with a white strain, he'd get sort of a pinkish strain, and then self-crossing the pink strains would not give a 1 to 2 to 1 ratio or a 3 to 1 ratio. It gave an interesting 1 to 4 to 6 to 4 to 1 ratio where it ranged from pure red to pure white. And what his idea was that you could actually use Mendelian principles, principles of segregation, and to discover, and independent assortment discover that multiple genes could contribute to the single phenotype, in this case, red color. If multiple genes showed incomplete dominance, you would produce phenotypes with a normal distribution. And that's the key for this lesson today. Multiple genes that show incomplete dominance will produce phenotypes with a normal distribution. Let's see how that works. Imagine crossing a red wheat strain with a white one. To represent the red genotype, we're going to use capital letters, big A, big A, big B, big B. To represent the white, we're going to use little a, little b, little b. They generate pink, which is intermediate in color, big A, big B, little b. Again, both are incomplete dominance. This is called the polygenic model or multi-gene model of genetics. So if you crossed two of these pink plants together, what would you end up getting? And you can represent this, of course, with a forked line diagram, right? Each one individually, the A or the B, would give you a 1 to 2 to 1 ratio. So simply cross-multiplying the 1 to 2 to 1 ratios against each other, you generate things that look like this. Here on the left-hand side is the A gene, 1 to 2 to 1. And then in the middle here is the B gene, again, 1 to 2 to 1. Multiplying the ratios, 1/4 big A times 1/4 big B gives you 1/16 big A, big B, or red. Notice that there are 4 alleles that all add to the red color, big A, and big B. 4 additive alleles is what we call these, and that generates the red. On the bottom of the panel, where 1/4 are little a, and 1/4 little b generates 1/16 that are little a, little b. None of those alleles add to the red color, so hence the wheat is pure white. So that's hence the reason this says 0 here. Now everything else is sort of an intermediate in between. If a wheat has inherited 3 of these big letters, 3 of these so-called additive alleles, the ones that add to the red color, it will be more red than white, right? If instead it inherits only 1, like for example this one, little a, big B, little b, it would be more white than red. And so there's a range of colors. Now we can graph these depending on how many of these additive alleles they have, whether they have 4, 3, 2, 1, or 0. And here it is. On the left-hand side are the red, pure red wheat variety. Remember, there's only 1/16 of those. On the other side, pure white, again, 1/16 of those. But the intermediate, the pure pink, has the most ways that it can be combined together to get 2 of these additive alleles. So again, here, oh no, I didn't put it on there. On the left-hand side, the red wheat has 4 additive alleles, 4 alleles that contribute to making the red color. Here, in this category right here, there are 3 alleles that contribute to the pink color, to the red color, sorry. And in the middle, there are 2 alleles that contribute to the red color. Here, there is 1 allele that contributes to the red color, and here on the far right, there are no alleles that contribute to the red color. The one in the middle is the most common because there's the most ways you can generate plants that have 2 additive alleles. Notice that the distribution looks more and more like a normal distribution, where the extremes, the extreme colors are the rarest, and the average is the most common. So we call this additive genes. These are polygenic traits, meaning many genes contribute to the same phenotype. Again, each gene, of course, has 2 alleles, one allele adding to the trait, and one allele that does not add to the trait. Alleles from different genes have the same effect. In the wheat example, gene A has the same effect as gene B. The amount of the trait is determined by the total number of additive alleles, regardless of which gene they are from. We call this quantitative genetics because the number of additive alleles are adding together to make the phenotype. There's a few assumptions that we're making here. One is that each additive allele contributes equally to the phenotype. This is almost certainly not to be the case in real life, where some genes contribute more than others, but it's a model, it's an approximation. For disease, the same thing applies. We all inherit certain risk factors or risk alleles for inheriting a disease. And some alleles are protective, and some alleles contribute to that risk. For example, cardiovascular disease. Some alleles will make it more likely that you'll develop cardiovascular disease, while others will help protect you from it. The combination of these additive and protective alleles determines your overall risk for developing the disease. Here's an example with diabetes. In the population, as shown by this normal distribution, we have a range of additive alleles, or predisposing alleles is what they're called here. Most of us will be here in the average, but there are a few people that are at the extremes, either on the left-hand side or the right-hand side. If on the left-hand side you have very few of these predisposing alleles, you would be unlikely to develop diabetes. Whereas on the right-hand side, you would almost certainly develop diabetes. Now, of course, with these kind of traits, it also depends on the environment. We're going to talk about that later. One of the other points I want to make from this graph here, though, is that with human disease, it's a sort of a false, it seems like a false Mendelian kind of principle where if you go to the doctor and the doctor diagnoses you with a disease, it's like you either have the disease or you don't have the disease. It seems kind of like an all or none. In this case, this is showing diabetes. If you are in this right-hand range down here, you have clinical diabetes. But in reality, there's a underlying normal distribution where most of us have sort of a predisposition to this, but you wouldn't actually develop diabetes unless you had a lot of those risk alleles. All right, so that is all for today. I will see you in class.